So good morning, everyone. I hope you're awake. We're going to go into the latest work on the research that we've been doing with a very large collaboration. We mentioned yesterday, I introduced you some of the people, some of them were in the room, and then we put that picture of the meeting that we have on Monday for getting gene-targeted therapies for MSMDS. And as everything, we start where we think the unmet need is the largest. And in ACTA2, you guys know there's a lot of mutations, but there are some that are much more aggressive that affect kids instead of adults. And that's why we decided to focus on this. And in reality, it's because the families have been asking us to do something. That's the true motor of everything we have deployed. I'll tell you very briefly while we get the presentation up and running, which is up there. It all started with, I told you last night, or for those that were in there, Cheryl and Seth and Avery were my first patient. She was 2 years of age. They thought she had mitochondrial disorder, but they tried everything, including a muscle biopsy. They didn't find it. They did a whole exome sequencing, which at that time was not a common diagnostics. It had to be done through research, and ACTA2 came up. And then when we look back at her MRI, which didn't have vessel imaging, but in one of the sequences you could see she had the classic features. And this is before we have the first paper from Diana saying MSMDS exists and is this mutation. And so we are kind of, you guys experience this in the community. There are a lot of families that have older children that have navigated years without a diagnosis. So at that time, on an afternoon, another scientist walks into my room and is like, Patty, you should apply to this grant. It's due by 5 p.m. It's 3 p.m. I have postdoc and say, Yamuna, what are you about writing this in one page? So we asked a company, that a Be Heard award is what we got. It's a little philanthropic award to make a mouse model. So we decided to make a mouse model. I contacted Mark, said, Mark, this is your realm of expertise. Mark is the expert in ACTA2. Do you wanna do it? And
I think I was the only one in the building who had heard of the condition.
Yes, exactly. And that's what brought us together, by the way, were the patients in the ICU. And in fact, it was when Raislin arrived to MGH and we had to troubleshoot what was happening and how could we keep him going, that a very big team of clinicians that happen to have labs in this hospital, it's very common, got together to start thinking together and thinking what can we do, what can be treated, what can be done. So we put that in place. It takes a year to get a mouse. It takes another year to see if the mouse does what you want. And what we're going to present, we haven't presented anywhere. The good news is that we have pretty nice, we always call preliminary data, okay? The truth is, until you trial it in a human, it is preliminary data, okay? So when you see something working, we're not talking about, if it was working in humans, you got a drug approved that the insurance has to pay, okay? That's to put it in brief. So it's always gonna be things that we're learning, things that we had to tweak. But what we are very excited with Mark to introduce today is that we have preliminary data that shows that we can do a very targeted gene therapy in this disease and really rescue the mouse model of this particular mutation. We pick arginine 179H because it's the most common across the patients. This can be done with arginine 179C and others. So it's not that you only have to do it with one, but you have to start somewhere to show that you can do the whole loop. And then we're hoping that we can expand in a platform way to other mutations that we can try to treat the cells in vitro of the patients without having to make another model and another model and another model. The good news also is that Diana Milowicz has a Ledoux collaboration of multiple investigators across the world to create models of mutations that affect aortic disease. So there's going to be a lot of resources to continue expanding a therapeutic gene-targeted therapy. So with that, I think we're gonna present this data, and it got accepted, and there is a couple of talks in a meeting that happens in the 17th to the 20th of May, is the American Society for Gene and Cell Therapy, is where all the gene therapy people come together, is in Los Angeles. And we've been selected to present in one of the platforms, nothing fancy, guys. like, just to present our results in that meeting. So they're going to be a full kind of out there. It's already out there, no? Like the abstract came out. So what we're presenting is mature enough to be presented also to the scientific community. We try to put it in a way that it relates to you guys. So you'll see a lot of human mouse. We're not trying to say children are mouse, okay? It's just to make the connections between the clinic. These are the guys that took us on. Like Mark and I started, and then we say, well, we need this, and we need this, and we need this. And you can see the expertise of these people, the institutions that cross. You hear Heather and all excited as we are, freak people, of having a lamb that is going to be ACTA 2, hopefully in October, being born, and that will enable things. Shout out to the group at NIH and Hopkins. They're experts in another genetic vasculopathy, and they're helping us because they're common pathways, and we use the same way to get to the cells. And you'll see that we will present work from all of them throughout the presentation, not so much from Carlos and Shira in this particular presentation. And what is this? There is an acquaintance of mine that I knew from a master's that this person did at the university, through other colleagues of mine that got affected by one genetic vasculopathy in his family, lost a baby at 6 months, and decided to do something and has the means. So the way that he thought he should do it is not philanthropy, is through something that can keep going, okay, that can grow it to be able to get to a clinical trial. So he founded this tiny company, is registered as a company, and through that company that's called Angel Biotherapeutics, he is funding these labs. Every single lab is also NIH funded. So I want to put that out because we're equally funded or more funded from NIH than this collaboration. And there is a lot of funding that we're going to need. It's not that we're done, guys, and there's not, we made it. There's going to be a lot of celebration in the sense that we're in the right track. But when you start having things that work is when you start talking about developing for a human. And that's when all things kind of go crazy in terms of cost. And this is what we have. We're in this particular building is Jim Gisela, the guy that described the first human disease gene, Huntington's, like the gene of Huntington's. And he started a center to try to see if we could do this. And that includes describing the disease. We talked about it yesterday at length, the identification of the gene that was done by Diana Milowicz, the mechanistics, try to understand really well how is it that from the gene defect you get a problem in the biology. And I love that we also had not just the vessels yesterday. We had for the first time the gut, the bladder, people thinking on how these mutations can affect those particular biology and organs. And then you start trying to see if you can get here, no? And then go back into patients with a clinical trial. So that's what we call the cycle. For those of you that haven't Googled a lot of gene therapy, or even if you have done that, there's 2 types of gene therapy at the moment in humans. There's more than 1,400 clinical trials in the Q4 of last year. So we are beyond that part that we were like 15 years ago when we did the first gene therapy for adrenal leukodystrophy, the other disease very deep to my heart because I work also with those families. But there are 2 types, and there's a lot of action going on. And if you're giving the therapy, the thing that is going to put the gene back or change the gene directly into the patient, we call that in vivo gene therapy. And you need a Trojan horse somewhere to put your thing that you want to deliver to the cells of interest that gets the gene into the cell. So I call it Trojan horse, is a nice analogy. And in general, most of these are AAV mediated, but now we have something called nanoparticles. So these guys are viruses. There's a kind of cold virus that is inactivated, doesn't do anything, doesn't multiply. They're really good at getting into your cells because they're viruses. So very safe in humans. There's hundreds of trials right now with AAV in place. And that's in vivo. And then the other type of gene therapy is ex vivo, which is basically we take cells out of the patient and then modify them in the dish, and then we put them back in. This is what we did with adrenal leukodystrophy. We needed to change immune cells. So we took immune cells, changed them, put the gene back, put them back into the kids, stop the degeneration of the brain. This is what you do in CAR T. So if you heard about CAR T and someone that has a leukemia and it's getting immunotherapy of that kind, that's also a type of gene therapy. So we're going to be presenting an in vivo gene therapy, which is, the hope is that we can put through the veins an IV, and I'm literally talking about an IV. We have right now, Canavan's disease is being treated with gene therapy IV. These are babies, and we just have them on the floor. We put an IV, and it's almost virtual that you're giving them a gene therapy, and it's having an effect, and we're very impressed with that. So you're going to deliver something. This something is the gene thing that you want to correct. So either something that corrects the DNA or something that is going to deliver the gene. In this particular case, we're talking about these CRISPR enzymes that are able, you remember Heather and Abby walking you through the enzymes that are able to change a single base, a single letter of the DNA. And that in this case, in this histidine that is changed from an arginine, you got to go from an A to a G to bring it back to be an arginine, okay? So it's called base editing because those are nucleotide bases, CAT, CEGT, and you're just putting it back to where it needs to be. And Mark is going to say more about it. This is a slide that I love. Mark made it, probably took him hours to do this pretty. And this is why MSMDS is so relevant, and that's how we're trying to pitch it, so people keep supporting us to do this. It's prototypical disease of severe smooth muscle cell dysfunction, and you heard it yesterday over and over. So how do we get into developing a therapy to get to the first in humans? As we know, we need to develop something to take this enzyme that is going to correct the DNA, and that's called vector, okay? So the AAVs and the nanoparticles are going to be called vectors from now on. So thanks goodness we have this guy. I don't know what we would have done with this room and all the technology. Thank you, Fran. So what is going on into that vector is going to be, this is an adenosine base editor. So you remember it's an A, the A is the adenosine, and you had to change it to a G, a guanine. So it's adenosine base editor. It's a particular type of enzyme. It's SP-Cas9, so it's a CRISPR enzyme. And we're going to use different other strategies also to try to modify these vascular smooth muscle cells and make them work again. So what is needed in the interactions that we have with the FDA? We talked about, Rebecca talked about natural history studies, and there are those that you look back and there are those that you look forward. When a disease is so rare, looking back takes usually many years of collection of data until you put it together. That's the Regalado paper, like 33 patients took many years to collect. And the standard of care changes because you know the disease, and you've been hearing in every session how we are doing things differently. No, like, oh, we're going to modify this and we're going to do that. So now the disease is not going to be the same. The kids are not going to have necessarily the same path, and there is heterogeneity in each patient that we need to understand before we put anyone through any treatments. And that's going to be the natural history studies that are coming. And then you have clinical trial interventional. They're both, all of them are clinical trials. If you're touching a human, we consider that clinical trial, okay? You can interventional if I'm giving you something to change the disease, and observational, it's just looking at how the disease progresses, okay? So, and then you got to develop all of this, and nobody touches a human in anywhere. I like my echo. It has an accent, my echo. How come? I don't have an accent. I have many. Depends on the day and the time of the day, the accent accentuates. It's working really nice.
It's the people online can't see it.
Oh, okay. You got it?
Keep going. Yeah, they're working on it.
All right. So those interactions with the FDA is what allows you have to have something called an IND, okay? Approved to put anything on a kid. And that has a process of approval, interactions with the FDA, and I'll tell you a little bit more of that at the very end. So we put ourselves, not ourselves, these two guys, there is more than 45 people working on the disease at the moment to really try to get a vector, okay? The Trojan horse is going to take the payload, whatever we do, which is the enzyme, into the right place in the body. Why? Because we don't want to send this everywhere where there is no problem. We will have to deal with the problems of the enzyme being in tissues that we are not necessarily willing to edit, okay? The ones that we want to edit, the gut, the bladder, the lungs, and the vessels, and the eye. So we got a vector in the mouse, okay? This is going to be important, that is really, really efficient at doing parasites, smooth muscle cells. The parasites are cousins of the smooth muscle cells, and also endothelium, but it's the first one that really tackles the smooth muscle cells in a more efficient way. When you look at the patients that are being treated right now and the approved gene therapies, the SMA gene therapy is AAV9. The approved gene therapy for hemophilia is AAV8, okay? So they have a number, is how they've been described. AAV9 is the one most used everywhere in humans. That's why we also using AAV9 always as a comparison of something that we know we could take to humans because we know the safety, more or less, of how that virus works when you put it in the IV, when you put an IV. Then these things, usually you get it to work in the mouse. To get it to work in a human is a really long shot. And that's why we want the large models, okay? That's why we need a non-human primate. That's why you need a lamb or a pig or something that has a bigger size, a distribution, and a closer biology, if you can, to human. So we know that we can do the same thing that we're doing in the mouse in a human before we put it in a human. So that's to be seen. And we have studies in non-human primates, and we're trying to see if we got the right viruses. What accelerates the translation is if we can also, you see, this is how we deliver this protein is fluorescence, and you can see it fluorescing the whole body of the mouse. So you put like kind of a camera and you look at the fluorescence and you can see it. And this is called, it's a promoter that is really potent. It's CMV promoter. It's a promoter of a virus, expresses everywhere. But if I wanted to get the heart, I better do a different promoter that only targets and can express that thing that I want, the fluorescent protein, in one organ. So you can start seeing, and the good news is these guys are very preserved across the species. So one other way to try to accelerate the way to get to humans and predict, the whole thing that we're doing when we're doing research is trying to predict how reliable, good, and safe something that is working in these models could be working in the mouse. And this is a little bit in one slide, the whole program. So we worked for years on this with Dr. Ben Klansteiber, which is the expert that designed the enzyme. So he customized an enzyme for us to get exactly to that place, to do it in a safe way, and to not get off targets. And then we take patient cells, and we also create a cell line, and Mark will talk a little bit more, and then we do the same correction and look at them, and you saw that there are many assays that we can do in the cells to see that the cells are going back to work, that they're back in function, and the FDA wants this in patient cells, okay? So they want to see that at the human level is the only step kind of closest to the human is the cells of the human outside in the dish. You also achieve the same efficiencies that you are getting in whatever cells of other animals that you're treating. Then we do the mouse experiments, and then we are trying to see if we can use a vector that is different, that is more specific for smooth muscle cells. This will take more time, and we may go to the clinic with not the best one, but there is always a better one a few years later, and at some point you gotta treat. So we're hoping to have a nanoparticle that really does target this. We would enable a lot of other treatment, not just MSMDS. And you heard about the sheep. So this is just to show you this vector. This is a slab of the brain of the mouse. Fancy here, kind of like schematic, but I loved it. And this is the vector. The virus looks like that. And now that you guys have a pandemic with COVID, everybody knows how a virus looks like. The spike protein. We have an expertise that wasn't there 10 years ago. We were talking to families about AAV therapy. So, you know, it's a virus. The thing that is inside the viruses usually carry their own DNA to make more of themselves. So we just use the shell. And instead of giving the viral DNA, we put something that we want to see. In this case, it's an enzyme that edits the mouse, and it allows you to see fluorescence, whatever that thing got delivered. And you can see that these are the vessels in the brain, and there's beautiful parasites, and we quantified all this, and it's under revision at the moment with the resubmission, so will be published very soon, hopefully, always fingers crossed. This was the most exciting thing to see. This, what you're seeing here is the wall of the aorta, okay, in the mouse. And this is the aortic root, the ascending aorta and the abdominal aorta. You heard there are different types of dissections, different parts of the aorta we need to treat. And this is how much you get of the layers of the aorta to become red or pink in this case, which is that they got the virus. And this was we were very excited about this because the other viruses that we have, AAV9 and 8, the most common ones do not give you this efficiency of getting to these cells and transducing them. And I think the next one is Mark.
Yeah, so this next work is presenting the development of the enzymes that have been optimized. And this is work of the Klemsteiber lab, who are real experts at this technology. It requires an expertise. I mean, I'm a molecular biologist, but this is more advanced molecular biology than what I usually do. But basically, in order to optimize a base editor, you have to have a sort of a workhorse. And so Ben's lab created a model of ACTA2 in a human cell line that's been used for many years in the laboratory called a HEK cell. It's a human embryonic kidney cell. Not as important as the fact that he was able to engineer it and have the R179H mutation with the most common cause of the mutation.
Testing.
So this allows, in a laboratory environment, you to test multiple combinations of base editors and PAMs and allows you to find a perfect place to put the base editor, because every single sequence in the genome will behave a little bit differently, and you have to optimize a base editor for where it lands. The site needs to be specific to that part of the genome, and then you want to minimize off-target editing, because these things are enzymes. They're just looking at DNA and changing bases. You don't want them randomly looking through the genome changing bases. There's different technologies like illustrated here on the right that you can go for, but we're interested in base editing because this is a specific monogenic mutation that is recurrent. So, sorry about that. Yeah, so Ben's work, this summarizes a ton of experiments that optimizing different base editors, and ones that also have been, the base editor enzyme itself has been mutagenized to have different properties and different efficiencies. And using that, we were able to, he was able to optimize a base editor that had a much better on-target editing efficiency. And that's illustrated down the bottom, that it's, as Patty said, it's a ABE or adenine base editor, this version 80, fused to the Cas9. And we're important for, and we'll get a little bit more into this in delivery, these enzymes are too big for themselves to be put into a single AAV virus. And so the way people have, they're just too big. It's the virus can only hold so much DNA. So the way people have got around this is to use, basically just to split it in half and then add a component that can help it find together. And then you have to use two viruses. So you basically have to deliver one payload, and then they find the cell. This has been, used, and that's what's illustrated on the right is the split version, so that you can load it into what we call a dual AAV. But importantly, we're able to get high efficiency editing on the left, the A to G, in both in vivo, but in cells. And then on the right, there's actually in vivo in mice with the dual AAV strategy. And I'll show you more about that in a bit. And Patty always likes to ground us and say, we have to do it in the patient cells. So these are actually in the patient cells that your children have donated to research. And so we've been able to take those cells from a skin biopsy, grow up cells, and then use the base editor. This is showing that in this disorder, the level of actin, actually the general form of actin, which is beta actin, is shifted in the cells in the children, and that's shifted from globular to filamentous is normal, but in the children, they have less F-actin. F-actin are the big stress fibers that help you contract your smooth muscle. And so this is just an experiment that shows that we were able, after base editing, to shift that balance back towards a normal actin distribution. So what we're going to show next is the trials, and at least the first in the mice. We wanted to simulate infantile disease first. And so what we did was had mice and injected them at day 3 of life. Day 3, mice live about 2 years, okay? So day 3 in a mouse is like an infant. They are small, they're still breastfeeding. And but it's a good time to test a developmental administration of a virus, and that's because of an AAV delivery system. And that's because as you mature, humans, mice, sheep, the delivery methods are going to change and may be different. And maybe you hit it right the first time, but you may have to optimize that differently for older children than for younger children. So in these experiments, the mice were injected retroorbitally, which is their veins are too small. And these were with dual AAV with our base editing construct, either AAV9, which is the standard, as Patty was explaining, versus this AAV-PR, which was developed by Casey McGuire's lab and Patty's lab in combination. So, of course, to test it, we needed a mouse model. And this was something that Patty and I did a long time ago, was make this mouse model. And it's really been, really informative. We've been able to use this animal model that really recapitulates the human disease and its complexity. So on the left here this is more from patient cells. So I was telling you before about how the F-actin is deficient in MSMDS. Oh, thank you. So this we know is a phenotype we want to track. In the mice, when you turn on the mutant allele, they similarly both, they keep expressing their alpha smooth muscle actin, but they have basically disassembly of the cellular cytoskeleton, which is the basic problem, right? The cells won't contract. That's what we need the smooth muscle cells to do. The mice recapitulate a lot of phenotypes, including prune belly and intestinal phenotypes, which I'll show in a bit. And we got pretty fortunate in that the mouse model is really penetrant and really shows, at least so far, all the multi-system nature of this condition. When you make a mouse model, you never know what you're going to get. Some mice recapitulate some things, some do it quite well. We got lucky in that. We can see pretty much everything. And sorry, that was a duplicate slide. So I just wanted to, when we inject these mice, one of the things we want to do is say, are we getting to the cells and are we base editing in the mice? So this is a slide that basically quantifies base editing both in the heart and the liver, which are big uptake organs for us. And we're able to see that the editing, we did get editing with AAV9 in these organs, but even better editing with the AAV-PR, which we're excited to see. So one of the things that I work on the most is the aortic phenotype in our animals, and the mice do have aortic dilation. We can do echocardiograms or ultrasounds of the mice hearts and the mouse aorta, and we measure it, and you can see it grow over time. So I'm a mouse echo expert. Actually easier than children because children wiggle. But we get a nice phenotypic change that's similar to the phenotypic change in the children, and in other people and adults, which is downregulation of some of the other contractile elements in the smooth muscle cell.
At 6 weeks.
Yeah, absolutely. So the aortic enlargement phenotype is pretty young. We echo them exactly as Patty was just mentioning, 6 to 8 weeks, which in a human, this correlates to school age and then adolescence. So we know from the work of Dr. Milowitz and their group that this is, from the aortic phenotype, this is really one of the strongest predictors of lifespan in children, and it's different than other folks with ACTA2 mutations. The R179 mutation, in the first natural history study was an average of about 14 years of age for either an aortic dissection or the need for aortic repair, which is a proxy for that. And this is the same type of repair that, in people that Dr. Cameron discussed yesterday. So we were pleased to see in the mice, and these are again injected at P3, that of this cohort where we actually injected the mice really early, we let them live till 8 weeks and then sacrificed them so we could look inside the tissues. But even at 8 weeks, the treated mice did better in terms of survival than the untreated mice. And I'll show a little more about this. We don't think this is actually aortic mortality in the mice. We think it's actually GI mortality, mortality from the gastrointestinal tract. And then we also have not as many animals, but we're getting more, which are long-term. So we're letting the animals that we've treated with this base editor live. We'll see how long they live, because that's the data we want. And versus the untreated mice, they really are doing better, even out to 25, 30 weeks, which we haven't seen any of the untreated mice survive. So this is the data quantifying aortic size. This is just a diagram of the aortic shape. And this is what we do in clinic, this is what I do, is we pay a lot of attention to the aortic size, the proximal aorta, the aortic root, and the ascending aorta. And we do that, and then in the mice, so you can see the untreated mice have a little bit larger aorta than a wild type mouse. That's the same thing in kids. It's not that they have these huge aortas. Their aortas, in Marfan syndrome and in MSMDS, they're all about 10 to 20% bigger. And then it's later when they start to grow much larger. But in the animals, just like in the younger children, it's just a little bit bigger, but the treatment, we're happy to see reverted back to a normal aortic size, actually with both AAVPR and AAV9, both at the root and the ascending aorta. The other thing that parents report and that we wanted to test was intolerance to exercise in the kids. The mutation doesn't just affect your arteries, it affects your ability to do things. And so this is from Anna and Patty's work on actually quantifying your opinions about how the children do with exercise. And then we can actually test exercise in the mice. This is called a rotarod. It was mentioned yesterday, but you basically put them on a wheel and they run until they fall off. So it actually tests coordination and exercise tolerance. And then we can also do something called an open field test, where they just kind of run around and you use a camera and you watch how fast they just kind of, because mice that's what they do. They just run around. And then but you can quantify it. And our mice, have a deficiency in their ability to do both of these things. In the open field, they're just a little slower. I'll show a video in a second. But also in the rotarod, they fall off. They're not as coordinated and they can't run as fast. And in so this was quantified, the rotarod, the spinning one, every week in those mice I showed you earlier out to week 8. And the untreated mice have poor performance, but when you treat with with a base editor, you can restore performance and also performance in the open field test. And it has different parameters. It's things like how long the mouse went around and thigmotaxis, the way they move, anxiety. Thank you. This is behavioral. I gotta look it up. And then how much time they spend vertical. Mice walk around and then they do like a little stand-up thing that helps them sense their environment. And all these behavioral indices were corrected. Just to, do I do it with the big? Yeah. I do it with, oh, I gotta start how do I start is it down here? Oh, okay, yeah, boom. Okay, so this is a mutant mouse. It's smaller, they don't gain weight as well. And this is a wild type litter mate. So you can see, they're a little less mobile. They they just don't move very much. And they follow their big brother around. And so that's what we are quantifying in the open field test. And then this is a mouse that's been treated, an Acta2 mouse that's been treated with at P3 with the Cas9 base editor and his wild type sibling who doesn't have the mutation. And you can't really tell them apart. There's actually a stripe on one of their tails, so I can tell them apart. But I labeled them so that you guys could see that they really, it restores their activity. But the other thing probably just to notice that it also restores their growth. They're the same size. And that relates probably to improvement in the gastrointestinal phenotype, because this is as you all know, it's just a huge challenge for the children is the GI tract. This is data from, that was presented yesterday by Dr. Raham, but, we know in the mouse model that the GI tract doesn't perform as well, both in the kids. This is, asking questions of the patients and then in the mouse model. And slide's a little slow. Oh, it's going through the videos. We know that, and then also the urinary tract has obviously really challenges both in bladder performance. And then as we talked about yesterday, bladder performance impinging on the kidneys. And if you can't, properly get rid of urine, it damages the kidneys with hydronephrosis. And we see hydronephrosis in the mice, which is a nice phenotype, so we can see the impact of our changes. And then again, the other thing that we can follow in the mice is the emphysematous phenotype, or the lung phenotype that we see in the kids. We know that this is in several children, as we discussed yesterday, but also, we can see that phenotype and the loss of the type 2 alveolar cells. And, nicely, the treatment with the base editor, you look across AV9 and AVPR, really did help the hydronephrosis. It didn't cure it. There's still hydronephrosis here, but better than the untreated animals. An improvement in the emphysematous phenotype. And I think Patty put in capital here, but we don't see, at least, with our analysis so far, toxicity in the organs that you worry about, like the liver. This is a little gross, but we're all adults here. So, oh yeah, it may just play on its own. This is actually just after sacrifice. The bowel still works. This is a wild type animal, and this is kind of, you can just watch its spontaneous motion. Oh, no, I guess I can't do next. This is the mutant, and yeah, it's actually playing, but nothing happens. And this is the problem, is that you'll see there's a huge fecal load here that hasn't been cleared. And this is a video that doesn't move. And that's the root issue. And then this is a treated animal. Again, not quite as vigorous as the wild type, but definitely it's moving fecal material through, and there's not a giant load of fecal material in the cecum, in the colon. So at this point, I'm going to pass it off to Patty to talk about what we saw in the brain and the neurologic system.
Thank you, Mark. And this one is in one slide where we talked about it yesterday on the more like an hour cerebrovascular. But we talked about the vessel disease causing in the brain, white matter injury, abnormal blood-brain barrier. This is how your blood stays away from the brain and only exchange what it needs. And when that's not happening, you get leakiness, and there could be toxicity from having things that the brain is not supposed to see. And we have evidence of that on MR perfusion and neurodegeneration. Basically, as the disease progresses, it impacts the ability to cognize. And then the biggest fear that we're trying to prevent at all costs is the large arterial ischemic strokes will take and accelerate the neurological injury. So in our mouse model, this animates, great. In our mouse model, and you got familiarized with this. So we have in an angiogram of an ACTA2 patient on the left side is the human, on the right side is the mouse, as we've been keeping so far. And here you can see a patient without MSMDS and a patient with this exact mutation, and the features that we were describing are classic of the disease. And when we look at our mouse, we saw both the distribution of the vasculature has less tortuosity. You may wonder, why is it so straight in the mouse? And the answer is pretty flat brain. You don't see all the folding of a human brain because it's a mouse brain. And it's this big compared to this big. It's like less than 3 grams compared to a 1,500 gram brain. So that's an expected anatomy. And what we're seeing is abnormal branching, straightening of the vessels in every single anterior and posterior circulation. And when we quantify it, and this is Indian ink, is one of the most standardized ways of quantifying beyond the MRI and the CTA, which these animals, by the way, are undergoing at the moment, as we were able to see on the MRI sequences that we have even to the level of capillaries. So we will have a lot of more resolution of what's happening with the mouse being alive, because for this, we have to sacrifice the mouse and look the brain from the outside. But you can see the same areas where we see critical stenosis in the human to be also affected by the mouse. And at the histological level, when you look, and this is a patient that Diana, in fact, she started the community, if you ask me, donated her autopsy and to research, and this is what was possible at the time. This is a vessel in the brain. It's called the middle cerebral artery. It's one of the most important vessels to feed like 2/3 of each hemisphere, each side. And that's what it should look like. It should look like with a wrinkle. After you fixate the tissue, you take it out, you put it in these little slabs. And then when you fixate, the smooth muscle contracts and wrinkles this thing here, which is the elastin of the vessel. So this is normal. This is the protein that is affected in ACTA2. Is in fact ACTA2 protein. We call it alpha smooth muscle actin isotype 2. And you can see the staining of that. And the other thing that you can see is this, what was shown also very early on in the mechanism of intima hyperplasia or proliferation, is they make more of themselves and they start coming into the lumen, narrowing the lumen of those vessels. Both seen in this like conventional histology and when you stain, and you can see that there is disruption of the layers of that vessel wall. And when we look at our mouse, irregardless of the size of the vessels, so these are in the choroplexus, in the fourth ventricle, in the hippocampus, you can see that they have, instead of thin wall vessels, they have very thick wall vessels. There are more cells, more nuclei, and they're narrower in their lumen. And when you look at the SMA staining, this is the mouse, this is not human, and that's the exact same thing. When you fixate or wrinkles, and this is what happens in MSMDS. These smooth muscles are so deficient in contractility that not able to react to that fixation artifact that we see. So they're pretty flat and they're smooth. And these are hippocampal arteries, a different caliber, and what they look like in the mutant, and what they look like in a control normal mouse. And this is the effect of the gene therapy. This was one of the most surprising things for us, was to be able to restore the wrinkle. And this is in the anterior and posterior circulation. I just took 2 sample arteries, but there is a lot of millions of pictures of these things that we have to quantify. And basically with both, with using the AAV9 and the AAVPR, we were able, through the base editing, we also look at sequencing of these vessels, and we do have editing in these smooth muscle cells, and they're starting to work and contract again, not only in the large vessels of the brain, but also in middle and small vessels of the brain. The function of those vessels is restored. So since it sounds like we can get the vessels to work again, what does it mean for the brain itself? So we were saying the brain really highly depends on this thing called neurovascular coupling and blood-brain barrier to be working, to develop, to be normal, and to not have dysfunction. And as I told you, in patients with this mutation, when you do put gadolinium in the vein, you can see it leaking inside the brain. It shouldn't stay in the brain. It's very subtle leaking. It doesn't cause edema, okay? But it's very subtle, but we can pick it up very consistently. And it's not only in the areas where the white matter was affected, so it's not white matter that is injured. It also happens in the white matter that is normal. It's truly the testament of the dysfunction of these pericytes, which are the cousins of the smooth muscle cells, express alpha smooth muscle acting, and they're getting deficiencies on their job, which is to keep the blood-brain barrier working. Another cell in the blood-brain barrier is the astrocytes. There are 3 cells, endothelium, pericytes, and astrocytes. And the astrocytes in the mouse, which is, I'm going to show this is blood-brain barrier. I mean, let's do this one. So the leakage, instead of putting gadolinium here, we also have it ongoing on the MRIs, but here we're doing a technique called Evans blue. We inject something that is blue, and it makes a control mouse. If you put blue in the veins, it doesn't show up in the brain. In an MSMDS mouse, if you put blue, you get a lot of that to be leaking, and it's leaking around the vessels as we are expecting that to happen. And the other cell type in that blood-brain barrier, the astrocytes, are very activated, and this is another surrogate of that dysfunction of the microvasculature in the brain, which is what the MRI can read. So we focus on this because they're biomarkers, a famous thing that we've been talking. How do we know if we have an effect when we go to the human? How do we know who to treat? We're trying to characterize biomarkers that could tell us that we are restoring function, and the function is meaningful and it has a reason to be addressed. So what does the gene therapy do? This is an untreated mouse control. This is an untreated mouse with the MSMDS, and you can see a lot of more brown. This is the hippocampus where your memory consolidation and a lot of activity happens, very highly metabolically active, a ton of vessels, as you can see here, a ton of extra GFAP, which is the astrocyte protein. So we call that activation. And then you can see that these 2 guys treated do look much more like a control mouse. So if you get early in the disease, we are able to also restore the function of the brain vessels, and we also able to get a type of inflammation that is very unique to the brain. The inflammatory cells in the brain are called microglia. They have their own name. These are the cells that we treat in other diseases like the ALD disease. This is what we're trying to correct. And that's why we do ex vivo cell therapy. We try to get cells from the bone marrow to make more of this. But you can see between the control and the untreated that these guys are activated. They're a lot more like stained with something called IBA1. And then again, treatment with AAV9 and AAVPR seems to restore that or diminish that neuroinflammation that we see in the ACTA2 MSMDS model. Again, going from the human, in terms of, are we making any difference in the white matter injury? This is the white matter of the brain. I presented this MRI yesterday. This is a histological correlate of that from the same patient that we have pathology and has been published. There is degeneration of the white matter. Those are the classic injuries. You see less stain in where there is damage. And you can see here, this is a control, and this is different types of stain, MVP and look so fast. And then you can see how the white matter on the mutant mouse is really diminished compared to the one that is healthy. And you can also see those holes that we also see in the MRI, these black holes that are perivascular spaces. We didn't know it when we described it in the neurology paper. We just described it cystic-like. Now we know there are perivascular spaces by the histology of the patient, the histology of the mouse. This is a patient, this is a mouse. We quantify that, and it's dramatically affected. So the white matter is at risk, and I think this is another thing we need to understand in the patients. Not every patient has the same burden of white matter. They all have white matter injury, but we need to understand what we can do to prevent more and to diminish the progression of that. So again, a wild type control mouse, a lot of this brown means a lot of white matter. The untreated mouse with MSMDS, a lot of less white matter protein. And then you can see this one has a lot of artifacts, and things are never perfect. Two treated half brains in this case. Why is a half brain and not a whole brain? The other half went for sequencing and mRNA and all this stuff. So we take a half for the histology, another half for all the molecular changes that we need to check. So we got restoration of myelin function too, which was, there is no such a thing in any disease in mouse either way. In vascular dementia, we are not yet, or Alzheimer's, or any of these degenerative brain disorders, we've never been able to make such a difference in any mouse model. With regards to what does it mean to the neurons, the ones that do the work of thinking and doing, we do know that there is, over time, these patients were beyond 30 years of age when she passes away, and you can see there is cortical thinning, and there is white matter volume loss, and there is some of the aspects of this is a stroke that is happening at the time. And we did ask that survey of developmental delays, and we do see that kids being affected. When we look into the histology of the mouse, we do see that neurons are not happy. They're less amount, they mean they are degenerated, and they do have features of degeneration. And when we look at the functional imaging, so this is a live picture of looking at blood flow going into the brain with a window, and the mouse are just sleepy, and we analyze them over weeks. And this is a variation of the most severe model, so we can let them live longer. And you can see how the motor area and the sensory are more affected than other parts of the brain, and that's because they're highly metabolically active very early on. And this is basically that picture in motion. So you're basically sensing the changes in hemoglobin by optical imaging, and this is what you get on an MSMDS mouse model. Much less autoregulation and neurovascular coupling. This, I think, is a very good story to the next talk, which is critical care and anesthesia, because this is what we're trying to see. If we have a condition like this that may be happening in humans, we really need to be very careful not to decrease neuronal activity, because this is already at a very critical balance. So we challenge that. There is possibility of going through the threshold of ischemia. And more fancy stuff that directly correlates to functional MRI, again, something that we can do in the kids and the patients all the time. And this is the summary of all that we show you on the research side. We were able to develop a model. We got really lucky, as Mark was saying. The other disease we did gene therapy on, FDA approved, we never got a mouse model. Okay, and we had to do it with human cells, and it was a ton of risk, and we took it, but it is much better. We do have a mouse model now to test a lot of hypotheses, understand mechanism, and de-risk the therapeutics. We have been able to develop a better vector at the moment, and these POCs, proof of concept, that you can edit and cure the mouse. We got it, and that's why we're starting to interact with the FDA. That doesn't mean that in 6 months you have a trial. That means that we had to inform the FDA about the disease, okay? We have to tell them everything we know and what we want in theory to treat, and what our mouse model is doing and what we can modify. Follow that against another meeting with the FDA that we had to put a package, pre-IND, and then an IND. That's because it's a new drug. If it was a repurposing, much faster. And that's what we're doing in parallel, as you guys heard yesterday, with the small molecules. So far, the safety profile appears to be favorable. Both, we don't see off-target edits that are relevant or in big amounts. We're doing the off-target right now on the patient cells. It's something mandated by the FDA to make sure that we're not editing human genome anywhere else than where we want it. And this is where basically we're going with the data that we have of our cross-sectional, means the retrospective, when you just stop at one point and we look between what is known in the cerebrovascular and aortic phenotype of this disease. That's called Interact meeting in the FDA. When we complete a more thorough evaluation and we know exactly what to target and in who to target, and I'll tell you a little bit more about that, we go to the pre-IND and we hope to then, this is usually 6 to 12 months later, you manufacture and you go directly, and if they like everything, they let you treat a patient. And in the parallel, and this is required for rare diseases, okay? You do a natural history. Why? Because we're improving a standard of care. So FDA, if one day after all these experiments in the human is gonna give us approval, is because it's better than a standard of care. You guys get it, no? So there's gonna be always a parallel arm of people that do not qualify for X or Y reasons to get a gene therapy, to be monitored and contribute, to be able to say, we do have in the current times an effect that is significant and relevant. That's been the standard for gene therapy for rare diseases as of now. And these are very expensive things. We are gonna need industry partnerships very likely. And the whole idea is that you one day get a drug that you can give, like SMA. If you have a diagnosis or you know you're expecting a baby with SMA, you start moving the wheels and you get gene therapy in the first 6 weeks of life. And that's what we have available right now, a standard of care. But it took 10 years to get there. So don't desperate. It's a long journey, but we have good signal that, and we have people engaged. I think is even more important, including the people in this room. That is the most important. So we try to explain to the FDA, what do we want to do? We have to focus. And we know there is an early stage where cerebrovascular disease is more affecting the kids, like the strokes, the white matter. And we know in the second decade of life, we need to treat the aorta, or otherwise we start seeing dissections. So we put, like we call it stages, and then stage 3 is that decline that can happen as the kids lose function. And you guys hear us, we're gonna hear in the afternoon of older patients and how they're doing and what we're trying to prevent. And this is just to the note that in these rare and ultra rare conditions, each patient that is in natural history and being observed, the FDA has allowed us to do this. It is, we are gonna tell them what we're monitoring, and if the patient has changes in that, the aortic size, the cerebrovascular narrowing, the gut motility, that could become what we are treating on a therapy the next few years. So usually, at least depending on the disease and how severe and how rapidly progress, and MSMDS is not a rapidly progressive disease, guys, in most of the phenotype, okay? So we're gonna need 1 year, 2 years, 1 year minimum of monitoring to be able to say, this is what really is happening. And even the narrowing in the brain, more than half of the patients don't have it, okay? Until they are 5 years of age. So there is 5 years where we can watch what is going on, who's going fast, who's doing this, so we can catch them before they narrow those vessels. So it's a very dynamic design. It's a very complicated thing. We're gonna have to engage very expert people on these type of clinical trials to execute them. We are already, good news, on this is what a clinical trial looks like. You decide who can be treated first. You always have to focus on a specific population. You give the medication, and you said, the medication will be considered successful if you meet the primary endpoint. And you make this one, great, they give you approval. If you didn't make this one, this may rescue you, and you're gonna need a lot of natural history to support your claims. And that's why we want natural history to also occur in parallel. And as Rebecca was saying, we want you guys to be part of those contracts on how the data will be handled after the embargo, okay? Because we don't know where lives take us, but we know that the community will continue to exist. And you guys have to recap and do this. ALS has done this incredibly well. They just have 3 drugs now FDA approved for ALS. The first gene therapy last week approved. They did it this way. They went to the companies that did any trial, any investigator that did any trial, give me your data. And they started to look at what they really knew about the disease, what could be modified and not modified. So we're waiting for approval for the IRB for the natural history. And we have a contract with this small, teeny tiny biotech to be supporting that clinical trial. The natural history is a clinical trial, but it's an observational one. It will be posted in clinicaltrials.gov, and you guys, anyone will be able to see it across the world. And we expect to enroll the first patient in August. The last patient will be enrolled probably May, at the way that we interact with people and how fast you guys are being diagnosed now these days. And 3 years follow-up is the minimum that we want. We will hope to continue following up these patients for a very long term if they are not treated. And the idea in principle is that we flip them from being in natural history to the therapeutics whenever we have them, either a small molecule or a gene therapy. This is possible thanks to a lot of infrastructure and institutes and people and expertise and wisdoms. And we are part in very different ways. Mark is part of the Broad Institute. I'm part of the CR&D, Center for Genomic Medicine, and the same for Ben Clanstiver, et cetera, et cetera. We can map everybody. Again, this is a picture of the Monday meeting where we were discussing how do we continue the research. I said this last night, but I wanted to end with these guys, and thank you, and take questions if there is any time, Warren.
Hey, real quick, for anybody who needs to ask a question, but might be far away from the microphones, if you just raise your hand, we'll bring the microphones to you, or you could just stand up just like he did and ask the questions yourselves. Any questions? Starting with you.
Yeah, thank you. Very encouraging talk. I suppose you need to suppress the immune response patient that you want to use a viral vector to do genomic editing. So there would be risks then associated with that, and it then makes me curious about what's the minimum that have to be edited in any tissue in order to restore the lost function. In other words, how little virus can you get away with?
What's your background?
Reviewer number 2. Yeah.
Your question is so.
Your question's really on point, right? That's what the FDA wants to know. It's what we want to know, and we're working on that answer. I don't think
we know a little bit better their tissues are easier to separate the vessels than others. This vector that we have, we show you the brain pictures, it only goes to the vessels. So we're not expecting to edit neurons. We don't want to edit neurons. We don't want to edit myelin. We don't want to edit any other cell. So in the brain, which is the best one that we can separate the vessels as of now, ballpark 7 to 10% of the cells are being edited. And that's enough to get to the wrinkle. So that's the only thing I can tell you.
Of the whole brain.
Of the whole brain.
Like if you counted all the cells in the brain, it's a small percentage of them. We expect that they're the ones with smooth muscle dysfunction.
The microvasculature.
With the microvasculature.
Yeah, exactly.
And we're working on proving that, unequivocally.
Which will mean that we have to sequence each cell separately. That's called single cell RNA-seq is ongoing at the moment to get to your answer. But this is what it opens up when you start doing gene therapy. You start really answering the most critical questions in biology. How much you need to have normal to do okay? And then if you're doing okay and you don't have normal tissue, what are the challenges you still need to avoid because you may not have a complete normal function? We are more than impressed and having to explain this question to a lot of scientists. It's like, how much did you edit? How could the mouse look so good? And I think the look so good, guys, is that we flashed through it. We treat it very early because we're taking development on our side, okay? So we treat, remember that each cell that is edited can now, because it's the DNA of the cell, will give daughter cells that are now normal. So the earlier we catch, and if there is growth, is in our side because we get more cells to be normal. If we go later, and we did it at 4 weeks, 6 weeks, we're just getting the very first mice out of that, the editing seems to be kind of as efficient, but the disease doesn't change that much. And that's a testament of also certain parts of the syndrome that get established. And if the aorta is large, it's very hard to reverse an aorta that isn't large. Is the gut shut down? Can we really get it to work again? Our aim is to, wherever these cells are, give them some extra function. So some of them can decrease the needs of surgical procedures or things that are really morbid, like the pain that you can get from perforations and things like that.
And that's also why gene therapy alone is not good enough. You need small molecules. You're still going to need for some patients to find other options. And so that's what the Dr. Milowitz's group is working on, and us as well, because this isn't going to help every single patient, even if we get to the end game, there's still going to be patients we need other options for.
To the immunosuppression question that you asked for, every time we put one of these AAVs in a gene therapy trial in a human, we're learning something. So, and the FDA does learn with you and changes the requirements of what they ask you to do before you can treat a patient. But just to give you a sense, the immunosuppression is pretty aggressive and primarily steroids that we use for asthma and all kinds of things. In the first couple of weeks, we continue with a little bit of immunosuppression for around 6 months. And then we're not, these patients in general, and this is just very new, guys, editing in the human in patients, there's only 2 clinical trials out there right now. One is about to get possible approval, accelerated approval. So we know it can be done. It can be done in humans. These are adults. But those patients are usually immunosuppressed for around 6 months, okay? And then much lower than the beginning. Because the virus goes away. The virus is like a viral illness. It will get deteriorated and there is no more virus anywhere. So the virus lasts very little. We're gonna have to see if the enzyme in your body that is gonna be there for a little bit longer needs to require immunosuppression or not. And these are the questions that we don't know yet and trying to learn from others that are taking the lead in the human trials to see what is a regimen. We develop a fellowship in gene therapy to train clinicians because who's gonna deal with this? Like, who's gonna know what to do with these patients? Like, we are having trouble to treat these patients in a standard of care. Once we do a genetic therapy or any kind of therapy, we're gonna need people that start knowing their thing.
Good morning. Angela from Sweden wants to know if you can explain how MSMDS affects the metabolism. Is it possible that MSMDS affects the thyroid gland?
We don't have evidence of, so we do check thyroid in every single kid, in every single patient. Anyone that has any vascular disease, thyroid disease is a risk factor for vascular disease, so we constantly on top of the thyroid hormone. I can tell you that we don't have evidence of disease on the thyroid. We do have kids that if the families have Hashimoto, they have had Hashimoto, like one that I can recall, but I don't think we have evidence of disease in the human on the thyroid that is significant and gives you alterations of that kind. Having said that, the thyroid is also an organ that has a lot of metabolic activity, needs vessels to feed it. Could it be that over time in the lifespan, there may be challenges? We don't know, but it's possible. Yeah.
I'd say that Dr. Murdoch presented evidence that at a cellular level, there's definitely changes in the metabolism of the affected cells. But that's different than the metabolism of your whole body, like in the thyroid would affect.
Hey, so that's amazing. Can you so you guys saw recovery of the white matter? And was that seen in MRI as well?
No,
hard to say.
I don't think we have data yet. All right.
Because we injected at P3, I would be guarded, and I would say we prevented degradation rather than
reversal.
reversal.
Yeah. Really nice segue to, we treated P14. These are like late teenage, young adulthood kind of stage mice. We see an effect. They're doing better. The dose that we gave them, not enough. Okay, so we went back, put the dose up and see if we can do better. Okay, but it's telling us exactly what I think is hinging here is there is a developmental aspect that we intervene early, we could prevent. That's prevention. Reversal is a different thing. And I think for any kid that is not an infant, we will aim for halting the disease. Like my best will be if we can keep the aortas from dilating. Okay, if we can keep the vessels in the brain from narrowing. In fact, for the vessels in the brain, I have a lot more hope because vessels in the brain of the smaller type remodel over the course of 1 or 2 years. Like, and the endothelial cell can last 2 years in the brain. So we're gonna have to wait. But I'm hopeful that if we give new function to this vasculature, maybe we get better vessels in the brain. For sure, vessels to be able to move the blood flow better around the brain. And that will be cognitively and functionally a lot better. That's what patients that get the revascularization surgery do feel when they get new vessels put like on top. They're not new, they're also of a like they're ACTA2 mutant, no? Like we got to remember this part. But even that, it makes them a little bit feel brighter and less fatigue. And that's usually the patient-reported outcome that you get from the EDAS surgery. So less headaches, brighter, less fatigue. So improving blood flow will have consequences, we expect.
So first, thank you, Dr. Musolino and Dr. Lindsay, for just everything you're doing for this community. I think this is really hopeful news for us. I have two questions from the lens of us lay people that are not doctors. And, if you could just, you put the timeline up there. Was that for the patient registry? I think what would be helpful to us as parents and caregivers is just to provide a timeline. Like you had said that the first patient would be enrolled in the patient registry in, I think it said August of this year, and then it would be 2027. Can you just give us a timeline to say, when would be the most best case scenario a gene therapy would be available, and just take us through that timeline a little bit?
We took that bet and put it in the graph. If you go down to the last one, don't do that. It's gonna drive you crazy. Oh, you had to exit presentation. So basically, we took the best guess at the speed that we're working, and I will say something. We haven't faced headwinds of the real kind in research, guys. I'm expecting them. I'm prepared for the 6-month delay, the 12-month delay, because we're getting too lucky, and that's law conformable. You're pushing, you're gonna get resistance. So I say this so you guys get prepared. But in the way that we're going, I put here this thing, pre-IND. You don't treat anyone, first in human, until you get an IND. So we're talking about 2025, okay, for any patient to be treated. Which patients are gonna be eligible will depend on what we get on the experiments and the long term and everything. What we get on the large animals. We do want a large animal study to know what dose we need, guys. Dose in a mouse. Let's put it this way. We cured the Marfan mouse how many times, Mark?
Yeah, it's a point I like to make, in rare diseases. We found lots of treatments that cure mice. And so, as you get bigger, it's a challenge. And so, we have to pass each of those points. We need to see that it works in a large animal before we contemplate children.
Yeah.
And it's harder. It gets harder as you go. But yeah.
So I think the other question, just as a follow-up to that, to help us as patient caregivers and advocates, is to just, if you could just quickly just highlight where the parts that we get back involved, like where will we be participating in this process? So just so we have a heads up.
There are multiple things going on in parallel, and I think it's important. The foundation and other foundations should get the registries up and running, patient-based, patient-owned. I really believe this is something you guys have to do. Like I tell you from experience. And advocacy groups are also not perfect. No, a family that is leading and moving and pushing. We get these guys here to have to take care of the kids because they are having things to slow down or have to halt. And you have to be able to, as a community, be able to claim this thing and take it forward. So in parallel, there is registry already and biomarkers studies at Houston in Dr. Milowick's lab. And you guys, David, could you circulate the slide of the contacts so we can print it out and give it here so they don't have to fish through the presentation. So if you have a printout. Excellent. Okay, guys, there's a flyer at the front desk. So that is going on. We are a lot focused on the cerebrovascular aspect. So we have a lot of other areas that we are looking, like MRIs and stuff. And we have a biomarker study too. The two of us have the ability, our study, both Lindsay and myself, that you can consent. You guys, all of you, I think, have consented to pretty much both at the moment. So they're all, those studies allowed to sharing, okay? So I can sell the cells from you guys to them. They can sell the cells to us. It's about putting agreements between institutions, things that take time, blah, blah, blah. It's absolutely possible. I'm letting you know this because that's the way we write our protocols. Next, we're funded by NIH. NIH demands that we put all the data that we learn after the embargo and make it public. Everything that we learn in these mouse models and stuff will go public on libraries, okay? So that's the other way to never lose the material. And another study is this one that you're hearing, which is going to be getting a lot of your standard of care, if you want to, in a very standardized manner. So we have the same intervals, the same scanners, the same people doing the assessments, and includes physical therapy, MRIs of the head, MRIs of the aorta, includes testing of exercise, pulmonary testing. It's like basically you come for a week. We do this in every single rare disease we're having gene therapy at the time. And we do it funded in a way that can be used as your standard of care. So you don't have to get the same study done later somewhere else. You take it, you can get a report, and your physicians can also report it themselves, which is something that allows these studies to lessen the burden of patients, no? Like if a kid has to come here to get a CT or something, we decided not to do CTs in our studies, all MRI based. So if you're doing this and you're putting the kid through traveling, the family, blah, 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 we want return. And the best that we can do is to give you everything we have acquired so you can take it to your local clinicians and share it and use it for standard of care.
And you could say, like, I don't need my aortic imaging this 6 months because we just did it and here's the results.
And here's the echo and here's the results. And they can put it in the chart. It's in Epic. It will be available across institutions too. Complex answer. There are like multiple studies. I want you guys to get engaged as much as your capacity allows you. On participating on the study that they have with Diana, you gotta make, they will be informing you about the entire design, which has her 2 months, there probably other aspects and what is included, a blood draw and all of those things, the same thing for our study. Guys, I want you to ask questions. We did a webinar. I put at the end one slide. I want you to ask those questions because that's what you need to know if you're going to put your family through something in research. If it's something that you feel has the risk benefit and the things that you need to get engaged. And then if it's a 2-month study and it has a follow-up and stuff like that, we encourage you to really pick. And I don't think it will be exclusive or we're going to let you out because you're participating in that one. We, on gene therapy, I'll tell you my experience on this. We excluded only patients that were on experimental drugs, okay? So if you were taking another gene therapy, if you were taking another drug has never been approved, we did not allow you to participate on the gene therapy, natural history. If you were on an FDA-approved drug that could be repurposed, okay, we were able to keep you in our studies. We'll have to check with our IRB and the people that help us design this to say like it's okay to put it in a vitamin or not and these kind of things. But I think my gut feeling is we can still participate in both if you're willing to do that. It's up on what is involved in each study, and you guys have to sit as a family and have a conversation with yourself and with your doctors, the one that you trust, your champion. Like for our consents for any of the interventional trials, mandatory that the patients have the consent, discuss it with their clinician, have 3 days to discuss before they can tell me that they want to participate, and we fly them to Boston. And then they come here and we have a 3-hour consent with 3 witnesses to make sure that you guys first are asking the right questions, understand, and want to do this. So you're driving, so you have to be informed to be able to drive decisions.
All right, we have 2 more questions online. Andrew says, great talk and terrific data. Both the AAV9 and the AAV9 PR seem to work. Will the trial therefore be AAV9? And the dose IV presume is going to be very high, but congrats on the very impressive reversal in the mouse.
So you wanna?
Yeah, sure.
Yeah.
So AV, there's different challenges to those 2 vectors. We're seeing better performance with the novel vector in terms of in vivo editing. However, that would need to go through as part of the approval process to treat a patient. So we need to generate additional data in order to use that particular vector. So since the AAV9 is working, it's still up in the air whether or not what the best path forward is, and will depend on future experiments. And is the simple answer.
And we're hoping that we get a ballpark of what is our pick, okay, by the end of this year. If things continue to go at the speed that they're going, we'll hopefully be able to say, okay, worth going with this one, not going with that. To the dose that we use is pretty much 10-fold lower than the IV doses of AAV that are being used for Duchenne muscular dystrophy, SMA, or anything. So this was much lower dose, and the benefit, and this was difficult to convey to any company. We talked to 1,000 people, but they didn't believe us. And I think the reason is we don't want to get into the tissues. We want to stay in the vessels. So we were able to get there with lower doses than what you need to get to the inside the brain cells, okay? And that's always a question we get with people that have experience in gene therapy. It's like, what dose do you need to do this? And to our surprise, this dose is 10-fold lower than the highest dose using humans at the moment. Means very little until we see a sheep or a non-human primate, guys. So again, more experiments to make kind of those kind of decisions. What is a dose of this therapy that could get you the editing that you need?
Thank you. And Angela asks, what should we do the families to help accelerate the process on getting the gene therapy for our community?
What you're doing.
Yeah. Keep networking and participate in the research.
And participate in research. And I would say it's an ultra rare disease. So you guys have complex kids, okay? So you have to, there is a much busier life on the back than a regular person that has just a full-time job and kids. So I will highly encourage, this has happened in almost every community, that you also participate in other foundations. Just make sure that the efforts are concentrated on you. You're too rare to be part of something. But just to give you a sense of what I'm talking about, a foundation that developed and helped get the natural history and the data for the gene therapy that got approved, ALD Connect, that was their mission. That's what they did. That's what they do. And they even got in front of the FDA for one of these patient boys to the next gene therapy for the adults, because they only treated the kids first, and now they need to treat the adults. Another foundation that was in that disease took on the newborn screening. And they hammered 6 years, every single Congress in every state, until they got to Congress to get newborn screening approved. Worth a mission, worth the work. It might be on your call. Is that what you want to do? And I know the Ritter Foundation, for example, is trying to get whole exome and like a study in one of the states to see if we can pick up these babies sooner. Like, can we just diagnose them from just genetic testing? I vote that we get that check mark of does your pupil constrict on something that is currently standard of care, has no cost, and is implemented in every single state, in every single country, not just the US. So if you guys can campaign on something and you want to join Ritter or any other foundation or make your own, do that, please. We need to get these babies to be diagnosed when they're born. They have an easy giveaway. Shine a light in the eyes. Tell me if the pupils constrict, and stop saying that these are newborn pupils. Like, a doctor can tell these apart if they're just having to check that mark. So that's my other pitch to the pitch that Dr. Murdoch says, that they're looking for volunteers for the writer. See where they're working on, see how you feel working with them, and try to see if you find a common mission aligned and the resources that you need to do what you want to get done. All right. I don't know if we have more time. Okay, so for the people virtually, Dixie is telling me that we will send a copy, electronic copy of the flyer for the participation in the studies in the Houston side. Thank you.